Thank you very much. So I'm really excited to present this work on behalf of our team, led by John, as well as Elizabeth Strakowski of the National Institute of Standards and Technology, and the JCVI team in particular, co-first author on our recent work, Li Sun, Kim Wise, Nasira Assad Garcia, Bogomil Karish, and Raywon Shaw, as well as Andreas Mershon and Neil Gershenfeld of the MIT Center for Bits and Atoms, and Mark Ellsman and Thomas Stiernick of UC San Diego. So for motivation, JCVI SYN 3.0 was minimized with the constraint of population scale growth rate. But when the team at UC San Diego prepared these electron scanning micrographs of the cells, there was a great difference in the morphology at the single cell scale, where JCVI SYN 1.0 had many cells that were about the same size and shape, roughly 400 nanometers in diameter, whereas JCB ISIN 3.0 included cells of very different sizes and shapes that all appeared kind of stuck together, suggesting some lack of control over cell shape or cell division. So questions motivating this project were, one, how can we make these images dynamic? Can we prepare movies to show how SYN 3.0 self-replicates dynamically to create this morphological variation? And secondly, which genes are required to restore a normal morphology more similar to that of SYN 1.0 in the genomically minimal context of SYN 3.0? So to pursue this, we adapted a microfluidic chemostat. About 10 years ago, I had contributed to a microfluidic chemostat for imaging the growth of E. coli bacteria, which grow as a rod shape, whereas SYN 3.0 includes a wide range of shapes and sizes. So Elizabeth developed a microfluidic chemostat, which has a similar working principle, but the chambers in which the cells grow are larger, and they therefore accommodate larger cells as well as smaller cells. A key point here is that mycoplasmas don't have a cell wall, so their shapes can be deformed by shear forces that are present in bulk solution. A major advantage of the microfluidic hemostat is the cells are shielded within these chambers and they're not exposed to flow. So fresh growth medium diffuses into the chambers, so the cells are provided with a continuous supply of food, but they're not exposed to flow, so the shapes that we observe in these chambers are the intrinsic shapes of the cells due to forces generated by the cells themselves, as well as confinement to a depth of 3 microns to facilitate imaging. And this is a movie that shows the growth of a genomically minimized strain in which we observe a wide range of shapes and sizes growing within this microfluidic hemostat. So using this platform, we did a control experiment and we observed that SYN 1.0, consistent with our impression from the SEMs, grows as small cells that are near the optical resolution of the microscope. But in contrast, also consistent with the SEMs, SYN 3.0 exhibits this abnormal morphology, resulting in a wide range of shapes and sizes. And using the chemostat, we can observe how those change over time. So, so now transitioning to a question of which genes are responsible. So similar to what Daniela Bittencourt presented during the first day of the workshop, as this genome is composed of 8 segments, it's possible to mix and match minimized segments with an otherwise wild-type genome. And as Daniela showed, one of those 8 segments was involved in um, what's the word? Starts with a P, when the cells can infect other cells. I'm sorry, I'm blanking. But in this case, we lucked out in the sense that the wild type, the minimized segment 6, in the context of an otherwise wild type genome, was the only minimized segment that conferred this strange morphology. And so therefore we were able to restrict our attention to the 76 genes that were missing in this version of the in this 1/8 minimized cell, as opposed to the 428 genes that are missing in SYN 3.0 compared to SYN 1.0. And then, so here's the growth of RGD6 in a microfluidic chemostat showing these strange shapes and sizes. And then this motivated the search for versions of a nearly minimized cell that contained additional genes in segment 6. So this is a movie showing the growth of JCVI SYN3A, which it resembles the growth of JCVI SYN1, where there are many cells that are small and near the optical resolution of the microscope. So Then turning our attention to so JCPI Syn3A retains 19 genes that are not retained in Syn 3.0, and they were organized in 8 clusters of contiguous genes that could represent transcriptional units. And when we looked at these clusters, we observed that the first cluster contained FTSZ. And FTSZ is a highly conserved tubulin homolog that polymerizes into a ring at the division site in most bacteria. So we thought, oh, if we add FTSZ back to the Syn 3.0 genome, then we could restore normal division. But to our surprise, addition of the FTSZ containing cluster alone did not restore normal morphology or division. The cell still had a wide range of shapes and sizes. And so this work was led by co-first author Li Ji Sun. And so Li Ji pursued a wide range of approaches to test in a systematic way the different genes that Syn3A retains compared to Syn 3.0. And based on her work, she discovered that 7 genes of the 19 are together required. Any one gene absent, and the cell reverts to that pleomorphic phenotype. And the exciting thing is that of these 7 genes, 5 of them do not have a known function, but based on their sequences, many of them are predicted to associate with a membrane. But the mechanism by which they shape the membrane remains unclear. So in the remaining minute, I just want to highlight our current work in which to guide experiments, we're trying to develop biophysical models for ways in which genes could shape the membrane and therefore contribute to cell division. So in particular, this is an equation that describes the bending energy of a membrane. So similar to the way a one-dimensional spring will try to relax to some equilibrium shape, this equation describes how a vesicle will try to relax to its equilibrium shape. And a key parameter here is this parameter called the spontaneous curvature, which is related to how the intrinsic curvature in the absence of any external force. So what's really exciting is that these kinds of equations, you can predict shapes as a function of the volume to surface area ratio, which is the x-axis, and then the spontaneous curvature parameter, which is the y-axis. And in particular, every point in this space represents a different shape. So for example, this red arrow from left to right, these are the shapes that correspond to that red arrow, where you go from this oblong shape. The idea is you have a vertical axis of rotational symmetry. So this would be like 2 basketballs, one on top of the other. This would be a kind of asymmetrical small basketball on top of a big basketball. And this would be kind of an oblong shape. So it's possible to predict these shapes. And in particular, for one cell, so in the microfluidic hemostat, we observe these qualitatively, we observe the different shapes that are predicted in these vesicle phase space diagrams. And qualitatively, over the cell cycle, this star corresponds to a single spherical cell. This star corresponds to 2 spherical cells that are joined by a thin neck, which we could imagine is the shape of the cell just before division. And this arrow represents the transition of the cell as it grows and then eventually divides. So from this, we can claim that the volume to area ratio must decrease for the cell to divide, and the spontaneous curvature must increase over time. So what are different ways that the spontaneous curvature can increase? There's a wide range. These are reviewed in work on vesicles. One is proteins such as FTSC. But within this framework, we can expand our perspective on different ways that the cell could do this, because we know that FTSC is not alone sufficient, and there's also data that FTSC isn't even necessary for the cell to divide. So different ways this could happen are small molecules such as calcium that can bend the membrane, polymers, perhaps the polysaccharide capsule outside the cell, which can cause the membrane to bend away from this polymer due to exclusion, or nanoparticles that can adsorb to the membrane and cause it to bend, perhaps such as the chromosomes. So this is all speculative, but I just wanted to highlight what we're currently working on. So with that, again, I'd like to thank the team as well as Jason Krausch at NIST for technical advice. So thank you very much.
Questions for James?
James.
Hi, Sebastian.
Great talk.
Yeah.
I was wondering, how are the 19 genes that were added to Syn3 to generate Syn3A, how are they selected?
So Syn3A was discovered in a sense so my knowledge of the history is that Ray had developed a series of deletion strains in an effort to further minimize the genome. And so there were, in the freezer, there were a variety of strains that included more genes than the fully minimal SYN 3.0 segment 6, an increasingly higher number of genes. So the SYN 3A segment was discovered in the sense that it included FTSE, so that was a kind of a hint. That later turned out to not be alone sufficient, but it was one of the smallest minimal versions of segment 6 that still conferred the normal shape. And then I think also to your question, so that's the identification of SYN3A, which was in a sense kind of an incremental deletion process by Ray and team. And then one other point about those additional 19 genes, Li Ji added those back to a different region of the genome. And so this was a way to test that it's not just those genes in their endogenous genomic context which is responsible for the normal phenotype, but rather inclusion of those genes in a different part of the genome could also restore normal morphology and division. So yeah,
Great, thanks.
thank you.
There's a lot of luck involved in finding SYN3A also, because we're still not entirely sure how it came to our interest. Ray has left our group some years ago, and it's something I should have a conversation with him at some point. More questions? James, do you think, you and I have, and our co-authors have speculated that we have recapitulated what primitive cell division may have looked like in SYN 3.0, and at least identified some genes that, while not necessarily sufficient for cell division, appear to be we showed to be necessary for normal cell division. But what do you think about how SYN 3.0 might be used as a model for investigating primitive cell division?
That's really exciting. So my first thought is, as SYN 3.0 grows fine at the population scale, even though it doesn't exhibit normal cell division. So that highlights that normal cell division is not a requirement for life. And John, you had also mentioned that ureaplasma parvum, for example, also doesn't exhibit normal cell division. So that's a wild type cell that doesn't exhibit normal division. There are also bacteria that L-form variants, such as L-form variant of E. coli that still lives, but it doesn't exhibit normal cell division. So in a sense of the evolution of normal cell division, the first point is, I guess, normal cell division is not required. That leads to the question of what's the fitness advantage of having normal cell division. My first thought is I'm reminded of work by Suk-Jun Jun et al. On bacterial physiology and the concept of a unit cell. So the idea is a unit cell is a chromosome and any proteins that are required for that cell to self-replicate. And if a cell can divide, as soon as it's able to make a new unit cell, and that's in that sense, the cell can produce more progeny that could then expand in terms of their range. So normal cell division might be a way to efficiently generate more progeny, even though it's not required for increase in biomass. And then, of course, my other thought about the primordial aspect is I'm looking forward to James Zaines's talk about the because I don't know much about the primordial life aspect. But those are my thoughts about that.
Thanks very much, James.